So hello everyone, I am Kexin. I'm really happy to talk here. So about our project scPNMF. So it is a sparse gene encoding of single cells to facilitate gene selection for targeted gene profiling. And so yeah, thanks for everyone who just contributed to the paper. Yeah, I'm really happy to present it here. And we recently uploaded our paper to our manuscript to bioRxiv. So if you feel interested after the talk, please just check it there. And so before we really go into details, I would like to first give a summarization on our method. So scPNMF, our method, it is an unsupervised method for single cell RNA sequencing data. And it has, I would say, two main goals or three main goals. The first one is to perform informative gene selection based on a dimensionality reduction algorithm called PNMF. So some key advantage of this method is that it can, so users can specify their desired number of informative genes. And this number could be relatively small. So scPNMF can usually just perform well, even with fewer than 200 genes. And besides, it outperforms some state-of-the-art gene selection methods in distinguishing cell types. And based on this gene selection part, we are also able to further project new data. By saying new data, we are saying, okay, so we can first select a subset of gene on a reference single cell RNA sequencing data using the first step. And then we can measure new targeted gene profiling data on the selected genes. And finally, we can project the newly measured data to a low dimensional space that embeds the same reference RNA sequence, single cell RNA sequencing data. And so that some information, some prior knowledge can be used for further tasks like maybe cell type prediction on the new data. And finally, because this is based on a dimensionality reduction algorithm, so it can also work as a dimensionality reduction method. But a key advantage is that it has good interpretability. So yeah, let's get started. So first, I will start with this dimensionality reduction algorithm, PNMF. So talking about dimensionality reduction, there are two popular algorithms, I would say, NMF and the PCA. So this is an illustration for NMF non-negative matrix factorization. So say that you originally have a matrix X, or the notation here is V, and NMF would learn those two smaller non-negative matrices, W and H, to give you an intuitive interpretation. So say that V is single cell RNA sequencing data. The rows are representing genes and the columns are representing cells. Then W learns a low dimensional representation of genes. And H learns a low dimensional representation of cells. Yeah, and some key advantage of NMF is that, okay, so some previous scientists have found the weight matrix W is really sparse. So it means that each column in W, which we call basis, each basis was determined by only a small number of genes. And the second advantage is that it gives a mutually exclusive representation. That means, okay, the determinant gene set are different for different bases. So that's the key advantage of NMF. And we will see why it is important in a few slides. And another very popular linear dimensionality reduction algorithm is PCA, Principal Component Analysis. The objective function is like this. And here, it removes the non-negative constraints, and it just replaces the cell, the low dimensional representation of cell H, that H, with W transpose X. And under this format, key advantage is that the weight matrix W, the learned weight matrix W, can be used to project new data. Say that you have a training data matrix X and you got W here, and now you have the measurement on a new cell, also measured on the same P genes. Then you can easily project this new cell to the K low dimensional space with this 4 W transpose times X. And it's very easy to use. And so this projection is a key feature of PCA. Unfortunately, in NMF, it cannot do the same thing. In NMF, if you have some new data, you need to factorize the matrix again. So it's not that convenient. And unfortunately, in PCA, it doesn't have the non-negative constraint. And thus, it doesn't enjoy the advantages of being sparse, being mutually exclusive in the representation. So here, some scientists designed PNMF, projective non-negative matrix factorization, about 10 years ago, to try to take advantage from both methods. So this is its objective function. So as you can tell, it is a combination of the previous two. So from the objective function, like X minus W transpose X, it is able to just do new data projection as PCA. And from the non-negative constraint, it also inherits the sparsity and the mutually exclusiveness from NMF. So the table is a summarization of those features for different methods. So as we can see, the algorithm PNMF is non-negative and enjoys high sparsity, high mutually exclusiveness between bases, and is able to do new data projection. So yeah, it's very good. And a question is, why do we need such a sparse representation? It is actually considering for some technology thing. Although right now the single cell whole transcriptome sequencing is really popular, but sometimes people can only afford to sequence a subset of genes. So like for technologies like RT-PCR, SC target sequencing, some spatial genomics like MERFISH, typically they can afford to sequence only up to 200 genes. And at that time, it is important to just design, biologists need to carefully design their experiment to decide, okay, which 200 genes to sequence on. At that time, so with the help of scRNA-seq data, if we can learn a sparse representation from those whole transcriptome data set, and then we can use this sparse representation to determine a subset of genes to sequence on. And then in those experiments, we just measure on that subset of gene. And finally, if we can match them back to the whole transcriptome data set, then it would be beneficial for downstream analysis. And this is exactly what our scPNMF is doing. It enables users to first select a subset of gene, and then it enables the users to project their new data back to the reference data set. Yeah, that's I would say, the main target of our method. Yeah, and let's move on to see how our method works. So this is an overview of scPNMF. As you can tell from the figure, it mainly contains 3 parts. The first part is running PNMF algorithm. The second part is about basis selection. And the third part is how we do like how we perform those applications. So let's start with the first one. So our input, the input for our algorithm X, it is gene by cell scRNA-seq data. We don't require users to do like complicated normalization. We are okay to deal with the original count or just to take log of it with pseudocount 1. Yeah, we just take a log count single cell RNA sequencing data matrix as the input. Then we run PNMF algorithm, and the output would be the weight matrix W learned from the algorithm. So it is P genes by K basis, and also the score matrix. So it gives a low dimensional representation on the N cells. And with the weight matrix and the score matrix, our next step is to do basis selection. Here, by saying basis, we are actually referring to the columns of W and the rows of S, the score matrix. So you could see, like, because there is a deterministic relationship between them, so actually they're referring to the same thing, and we call each one basis. And very similar to NMF, each basis here learned by PNMF contains positive weights corresponding to a unique small set of genes, and it does reflect, and this may reflect a certain biological function. By examining the data, we found some biological functions may not be relevant to, or may even obscure the signal of the cell heterogeneity of interest. So like, maybe for the cell heterogeneity of interest, that may be like the cell type. And we will show some examples later, and this is what we found. So because we need to be very careful about like the genes to be selected, so we would like to do a further basis selection to keep only those bases that has certain biological functions related to the cell heterogeneity of interest. So that's the motivation for this step. And how do we do that? We propose 3 strategies to perform basis selection. The first one is based on functional annotations. So basically you annotate each basis, and to just, if you found, okay, some of the bases may not be relevant to the cell variation that you truly care about, just remove it. And the second and the third strategies are data-driven. So basically the first one is we look at the Pearson correlation for each score vector with cell library size. And we would like to keep those that are not that highly correlated with that, because we would like to keep those bases that carries more information than purely cell library size difference. Yeah, so that's why we would remove those with too high correlation with cell library size. Yeah, and keep those carrying other information. And another strategy is to perform a multimodality test, like showing here. We would like to keep those with multimodality pattern, is that we believe it shows some subpopulation structure, and the subpopulation structure may be probably just the cell type variance. So we would like to keep them. And we combine all of those results and to decide, okay, whether each basis to be selected or not. And we only keep those selected bases from the weight matrix outputted by PNMF. Okay, and we got this called WS, the selected basis matrix. Okay, and then we go to the applications. Okay, sorry, on this plot, it is going from right to left. And so we start here. We start from the selected weight matrix. And as we have mentioned, for like targeted gene profiling experiments, many times the gene number budget is a major concern. So we would let users specify the gene number they would like to keep, and we denote it by M here. And we only keep those genes, the top M genes with high weights in the selected weight matrix. And those M genes are our selected informative gene. And with that, we do some truncation on the original data matrix and the weight matrix. Okay, and then we can do some downstream analysis for the first target, for the first task, informative gene selection. Okay, we just select those top M genes with high weights and truncate on the data, and then we can do clustering, visualization, and so on. And for another major task, for the new data projection, so yeah, we could project with help of the weight matrix. We could project the original reference data with the truncated weight matrix. We could also measure a new data set on the M selected genes and still use the truncated selected weight matrix to do low dimensional projection. And then, okay, so those sref and snew are on the same low dimensional space, but measuring different cells. Then we are able to train a model from the reference data and then to do prediction on new data. Yeah, that's the pipeline for our scPNMF methods. Okay, and then we move on to show you some results. So first, yeah, some people may ask, so PNMF looks really similar to PCA. So what is the advantage? Does it truly have advantage over PCA? So we will show you this. So scPNMF would output a basis matrix W. So does PCA. Usually people call it loading matrix, but yeah, they are just the same W we are referring in previous slides. So here, the first thing is that it outputs a more sparse representation of single cell RNA sequencing data. So telling from the heat map, you could see the W of PNMF is much more sparse and is strictly non-negative. And you could see like the gene clusters, like those with red lines are those gene clusters are more obvious. And yeah, it's just make it more easy to operate, easier to operate and to interpret. To have a look at its interpretability, we also perform GO-term analysis on the PCA weight loading matrix and the PNMF weight matrix. So you can compare, it's very clear. So for PCA, the biological process relevant to each basis, so first you see several overlapping between different bases. And if you have a look at PC1, so you can see ribosomes, like the cell division, like the immune system. So yeah, the biological process for each PC is not that clear. But for PNMF, the pattern is much better. So first you could see like each basis has its clear it has its own mutually exclusive relevant biological process. And if you interpret it like the first one is relevant to housekeeping gene, the second immune activity, and so on. So you can see scPNMF weight matrix is really more sparse and better at functionality interpretability. Okay, and the next step, the next result, I would like to show you why do we really need the step 2, the basis selection. So here in the panel C, the upper bar is about our second strategy, Pearson correlation with cell library size, because we would like to find those bases that carries more information than just cell library size. Then under that, we would choose those have a relatively low Pearson correlation. And if we set threshold as 0.8, then we select bases 2, 3, and 5. And for the lower panel, so it is showing the multimodality test strategy. So here we could see, if we set the threshold on p-value to be 0.05, we will select basis 2 and basis 3. And you could see basis 2 really gives very clear multimodality pattern. So combine those two, we will select basis 2, 3, and 5. And then let's have a look at how those bases affect the downstream analysis. So we draw UMAP on cells measured on selected basis versus unselected basis. So we can see with selected basis, the UMAP plot gives a really clear separation between different cell types. And for the unselected basis, you could see the pattern is not conveying any not conveying some true information. Yeah, so I think this is a result to show that the basis selection is really an essential step in our scPNMF method. Okay, and our next step is to show that is to measure its final performance on our main tasks, informative gene selection and new data projection. We first start with informative gene selection. So let's have a look at its performance in clustering. We measure that on diverse real data sets. There are 7 data sets. 3 are non-UMI, Dominus, Tirush, PBMC, SmartSeq, and 4 are UMI data, Gold, PBMC 10X, Gen4, and Gen8. So given a fixed number of informative genes, 100, we just measure on those 100 genes, and we reduce its dimension, perform different clustering, Louvain, k-means, and hierarchical. After clustering, we compare the clustering result with the given cell type label from the real data. And we measure the matchness between clustering result and the given cell type label. And the ARI is such a measurement, and the higher the better. And we rank all the methods in the order of decreasing average ARI. And we could see our scPNMF is the best. And it's worth noting that scPNMF actually beats some state-of-the-art informative gene selection method in some famous pipelines like SURET. SURET by default, it uses SURET VST, but we could see we are doing a better job. Yeah, so this is for clustering, and for visualization, we plot UMAP plot on the Gen4 data, also with that 100 selected genes. And we could see, okay, we our method is the first panel, we could see, okay, we clearly separate those 4 cell types, while other methods cannot do so with only 100 informative genes. Yeah, and it's an interesting finding that our method is consistently performing better across different number of informative genes. So we measure our performance on 20 genes, 50 genes, 100 genes, 200 genes, and 500 genes. And we can see our scPNMF is consistently doing better. Yeah, and so user may just run scPNMF based on the gene number that their experiment can afford. And yeah, scPNMF is consistently supporting that. Okay, and based on selected informative genes, we move on to do new data projection. And let's see how scPNMF guides targeted gene profiling experimental design. And finally, the cell type prediction. So yeah, this is a text description on how the prediction is doing. So how it's done. So yeah, so first we train things on a reference data set. Usually this is a single cell RNA sequencing data that you can get nowadays. You measure it on all genes, and we learn sparse projected matrix weight matrix from previous steps. And then we can map the original data onto the low dimensional space, and we can train the classifier, maybe random forest or other machine learning methods, and we can for cell types. And the most interesting thing is that for a new data set, we measure it on the only M genes we selected in the previous step. We measure it and use this projection matrix to project it down to the same low dimensional space. Apply F, we are able to do prediction on new data with help of the reference data. And to show its performance on real data, we generate 2 case studies from real data. And to compare with other methods, we still compare with those gene selection methods. So yeah, we first select genes based on different methods. And then we use PCA, the conventional PCA, to perform dimensionality reduction. And we compare the results between our scPNMF and those gene selection methods, like on different cases and on different the cell type classifier algorithm, like random forest, KNN, and the support vector machine. And yeah, we could see our scPNMF is always among the best. So the best is underlined. Yeah, so yeah, that shows our scPNMF really guides targeted gene profiling experimental design and may benefit cell type prediction. Yeah, I think that's all of the results we have right now. Yeah, finally, the same slides. Take-home message is that our scPNMF is a supervised method for single-cell RNA sequencing data. It can select informative genes. It can, select a limited number of informative genes. It can project new data based on the selected genes. And if you like, it can also work as a dimensionality reduction method for clustering, for visualization. Yeah, and yeah, finally, I would like to thank Dongyuan Song from Bioinformatics program and our advisor, Dr. Jing Jessica Lee, and thank you all for the help, in the project. Yeah, all of your suggestions are welcome. And if you are interested, please check our bioRxiv and our package on GitHub. Yeah, thank you. That's all for today.